All right, chapter 12, the cell cycle. So this chapter is going to be about mitosis. So mitosis is when the cells are going to divide. And there's three reasons why they would ever do that. And you've got them listed right here. Development, so when you're actually, a fetus and you're developing different parts. Growth, so when you were growing up into an adult. And then repair. So right now all we're really doing is just kind of repair. So cell division is going to be different in prokaryotic cells, which is bacteria, remember, versus eukaryotic cells, which is the type of cells that we have. So in prokaryotes, it's really simple because they don't even have a nucleus and their DNA is just kind of floating around. So if you look at this picture here, this shows you what binary fission looks like, and binary fission is actually how prokaryotes divide. So you can see what's going to happen is they're going to have what's called an origin of replication, and that is where the DNA is going to copy itself. That's where it's going to start. And what they're going to do is it's going to go all the way until it has two separate copies of DNA, and one copy is going to go over here, the other one is going to go over here, and eventually the cell is going to split into two. So it's a very, very simple process. You just got DNA being copied, goes to opposite sides, and then the cell splits. Now in eukaryotic cells, it's going to be a little bit more complex, and that's basically what the whole chapter is about. So if we go down a little further here, you can see that we're going to be talking about chromosomes. So our cells are going to have chromosomes, and that's how our DNA is arranged. So humans are going to have 46 of them, and they're going to be arranged in 23 pairs. So if you look at this one right here, you can see that they're all the way up to 22, and then here's the 23rd pair. And so you basically get one of each from your mother and father, and that's how that works. So we've got 23 pairs, which is a total of 46 chromosomes. So on your chromosomes is going to be where you find all of your genes, and they can have, like I say, hundreds to thousands of genes. Now, as you know, the number of chromosomes you have is essential to your development. And if you have an extra one or you don't have enough, then things can happen. And that's what we did in the lab when we were doing the babies, where you were doing those karyotypes. So when we talk about DNA, we're going to say that the chromosomes that have the DNA are going to be made of what's called chromatin. And so that's going to be DNA mixed with protein. So what I mean by that is if you look at this, nope, not that one, this one right here, the blue that you see here is going to be your DNA. And the little pink circles that you see there are going to be what are called histone proteins. Now, histone proteins have a positive charge, and DNA has a negative charge. So we talked about how that attracts, right? Opposites attract. And so what's going to happen is the DNA is going to be attracted to those, and it's going to wrap around them. So if you look here, we talk a little further down about chromosome coiling. So every 200 nucleotides, and remember a nucleotide is the building block of DNA, the DNA is going to coil around 8 histone proteins, and that's going to be called a nucleosome. So if you look at this right here, this little cluster right here is going to be a nucleosome because you've got 1, 2, 3, 4 histones, and then on the other side you have another 1, 2, 3, 4. So 8 histone proteins with the DNA, that's called a nucleosome. And what's going to happen is that is going to compact and compact further on itself. And this is a great slide to show you that. So if you have 1, 2, 3, 4, 5, 6, 7, 8, here's a nucleosome right here. So this is as zoomed in as we can get. Here's the DNA, and then here's the DNA wrapped around them. And then here's a bunch of them that the DNA is wrapped around. That would make one little piece of this kind of wrapped up stuff here. And what's going to happen is this is going to coil on itself like a telephone cord, and then that's what gives us our chromosome. And if you look at this picture of a chromosome, you can see how it kind of looks like fuzzy carpet, and that's because it's all kind of coiled up on itself. Okay, so let's go back to your notes. Now, I think I skipped a little bit here. Let's see. Okay, so karyotypes. This slide right here is an example of a karyotype. And all that is just kind of a spread of what chromosomes somebody has. And so if you have your 46 chromosomes, this is showing your karyotype. Now, chromosomes are going to have different numbers associated with them. You've got a haploid number and you have a diploid number. A haploid number is how many total, like how many sets of chromosomes you have. which in us would be 23. And the diploid number is actually the total amount of chromosomes that you have, which in us would be 46. You got 23 from your mother and 23 from your father. So 46 for us is our diploid number, and 23 is our haploid number. Now, if we look at the actual structure of a chromosome, it's going to have this kind of center part here that looks like the little waist of the little chromosome, and that's called the centromere. Now, what you're looking at here, I know it looks like the letter X, and that's how people usually draw them, but this, on this side right here, is an exact copy of this one over here. And they're joined together at that centromere. So they're called sister chromatids. So you have two sister chromatids together, and so let's say like this one is the one from your father, this one is an exact copy of the one from your father. Now if you look, oh boy, this thing's jumping around. If you look at this slide right here, you can kind of see that, right? You can look at this one, and then you can see there's an identical one right next to it. So that's, a copy of the one from the dad, and then you've got the one from the mom, and then a copy of the one from the mom. So that's what we're talking about there. Now another part that chromosomes are going to have are called telomeres. And telomeres are going to be at the tips of the chromosome. So right here and right here. And what happens is as we age, those telomeres start to deteriorate. So you can think of telomeres like the little plastic things on the end of a shoelace. And the older you get, those kind of fall apart, and eventually they start to fray. So that's a fun thing to look forward to as we age. Isn't that awesome? Okay, so we're going to stop right here and then do the next section.